Good morning. Can everyone hear me in the back? You know, if you want, there are more floor spaces out here. So my name is Shahryar Kianian. I work with the USDA ARS Agriculture Research Service in the cereal disease lab, just kind of next door from this building. And I'm adjunct faculty in the Department of Plant Pathology. Would like to talk to you about oat crown rust and the relationship between that and Buckthorn. But first, just to give you a background on ARS, if you don't know about it, is a research service arm of the USDA. We essentially, still confused on the buttons. We essentially work on problems of national importance. We are trying to do research on things that are important to the growers in the country, try to find solutions to those problems, and make sure those solutions are disseminated to the public, so at the end of it, provide economic opportunities for everyone to grow the crops they love to grow. The cereal disease lab, which is just next door, I always say look for the only building that has an American flag in front of it. Or if you're looking for a pot of gold on a day with rainbow, you can look for it. There is none in there, by the way. We checked. We work on diseases that are important to wheat, oat, and barley, the small grain crops. Many of those diseases tend to be rust, leaf rust, stem rust, crown rust. And we also work on fusarium head blight, which is a major problem in the small grain crops grown in the Midwest, and some in the southern part of the US. This research started long ways back, 1915, before soybean aphid ever made it to the US. And essentially was a cooperative investigation between the Agriculture Research Service and the Department of Plant Pathology at the University of Minnesota. And in the 1970s, we essentially moved to our own building, and we were called the Cereal Rust Lab for many years because we worked only on rust. But in 1997, fusarium head blight epidemic hit. It caused billions and billions of dollars losses to wheat, barley, and oat growers in the US. And essentially, that was added to our mission, and that's when we became the cereal disease lab. So the diseases we work on are the wheat, leaf and stem rust, oat stem rust, oat crown rust, barley leaf rust, and fusarium head blight. All of them nasty diseases, cause major damage, and essentially what we are trying to do is understand how those diseases progress across US, what is the variability for those diseases, and can we find plants that are resistant to it to essentially improve the crops. Why the rust? The rust have been a major problem in US for many, many, many years. None of us are old enough to remember that in the 1950s, and there were major epidemic of wheat stem rust or black rust, and essentially caused millions of dollars of damage in single years to all the wheat and durum wheat grown in the US. These losses still continue. With stem rust, there was I will talk about the alternate host. There was a barberry eradication program to get rid of the alternate host, but that essentially abated the problem. But it keeps popping up, and we also see this problem coming up with other rust, like the leaf rust that is in the US. So why oat? Oat is a major crop. US is the 5th largest producer of oat in the US. And the other part of it is that even though we don't grow billions of acres of oat, oat is popular as a free from food, free from allergens, free from pesticide, health reducing, cholesterol reducing effects. So it's a healthy crop that people love to eat. And as many of you have probably consumed it this morning, in some of your breakfast cereals this morning, they had some oat added to it. So it's a very nice crop, and you can see it's grown mostly in the northern hemisphere, but some in the southern hemisphere, in Australia and Brazil also, that they're producing lots of oats. Oat is decimated by essentially one major disease, which is the oat crown rust. Essentially infects the leaves, and you can see all the rust growing in there. And if it becomes really bad, the farmer actually plow their field under. And you can see on the back of this combine all the rust dust that is coming back in there. The reason these diseases were called rust, because in the old days when people walked around with their jeans or pants inside the field, they would come out and it would look rusty, essentially red. And that's why they were called the rust. And you can see the rust on the back of this combine just blowing off. This is what the heavily infected leaf looks like. And this is from Louisiana, essentially. And this is not red clay. Actually, you can see it's actually white clay. All the rust that is blown from the infected oat samples onto the ground. So you can see all of this oat is heavily infected with rust and is on the ground. You can try to use pesticide to control it, but remember, oat is grown as a healthy, chemical-free crop. So many farmers are hesitant about using chemicals on oat. But if they use it, they can do some control, but it depending on what time of the year and how heavy the infestation is. So oat crown rust causes major damage. As 2014 yield estimates essentially said it caused 50% damage to the oat yield in Minnesota and 35% damage to the oat yield in South Dakota. So you can see that is all of this rust, including the oat crown rust, are causing major damage to some important export commodities, wheat, barley, and oat. One of the ways oat crown rust spreads across the US, and all the rust spread across the US, is windborne. And this was known early on. If you look at this airplane, you can know how old that was probably. And essentially what happens is that the rust overwinters in the Gulf Coast or Mexico. And essentially with the wind, it gets blown up. And as the crop starts growing in different parts of the US, you go from winter wheat and winter oats to spring wheat and spring oat. The rust essentially ride the wind and come up north and infect the crops as they move north. So that's one way that the rust gets spread. Another way the rust gets spread is through their alternate host. So you have the life cycle of rust. You had the life cycle of aphid. Now you have the life cycle of rust. Essentially what you have, the rust can go through an asexual phase, no sex, just keeps reproducing on the crop plant that it infects. So in case of oat crown rust, it keeps reinfecting the oat, keeps reproducing asexually, produces more and more and more, and keeps on infecting. But when it comes to the winter time, it can overwinter in alternate host, in case of stem rust barberry, in case of oat buckthorn, and essentially go through a sexual phase in the spring. And many of you probably have seen barberry leaves with red dots on them. That's essentially oat crown rust during the spring. That is going through a sexual phase and generating more rust that can come back and infect the crop. Now, as you all know about sex, it causes variability. So you can look at your children, your brother's children, your sister's children, your uncles, aunts, kids. You know that the kids kind of look like a mixture of the parents. So they create lots of variability in there. Otherwise, we would all look the same. And that genetic variability causes more virulence in rust. So you can essentially create more variation in the rust that can infect the oat, and this keeps on going. So what happened in the 50s when we talked about the black rust or the wheat stem rust, essentially causing so much damage, there was a whole effort in the country to eradicate barberry. And people were putting salts and all kind of chemicals and things on their barberry bushes. There were even flyers printed out that, you know, if you have rust, you're a friend of Kaiser, you got to destroy it. And you essentially got the National Rust Busters Club too, that would give you awards for busting the barberry. So that was a major effort to essentially remove the source of alternate host for stem rust. It was effective. It removed lots of barberry, even though some can be still found in southern part of Minnesota and northern part of Wisconsin, closer to the Minnesota border, around the wheat fields. So there is still some barberry that can get infected with stem rust, cause variability, and infect the crops as it grows. All the new barberry varieties that are released by nurseries have to be tested to make sure that they are resistant to stem rust. And we have a scientist in our unit, Dr. Eugene, whose group essentially has one of the components of their project is to test all of the barberries that are going to be released in the US. But there are multiple alternate hosts. There are some that haven't been identified, people haven't looked for them, and we are essentially in search of finding alternate hosts, to finding what is out there that can cause genetic variability in the pathogen. The major problem, and this is the part that hasn't been addressed, is essentially studying the alternate host. Are there variability in alternate host? Are there any genetic variability in it? There are no genetic studies or no genomic studies. National Science Foundation, I don't think, would fund the Buckthorn genome project, but it would be important to find out if there is variability in Buckthorn for infestation to rust. The pathogens all have been sequenced, or almost sequenced. We understand their genetic structure, and we are using those information to studying their population structure. What is the variability in the pathogen population? How is that variability generated? Is it just the one that keeps blowing up every year, or is it the alternate host that is causing the variability? What are those things? With crop plants, wheat is sequenced, barley is sequenced, oat is almost sequenced, and people are essentially identifying rust resistance genes, identifying markers for them, so breeding programs can use them to essentially pyramid those rust resistance genes. With oat, I'll mention later too, there's over 100 single gene resistance that have been identified in all of the wild oat species. So what do we do with buckthorn? You guys all know it's a noxious weed, alternate host for crown rust. So what we have used, so what we know is that the oat crown rust pathogen is highly variable. This was a study that was done over 3 years with all the samples we collected from across the US. Part of our mission is essentially understand pathogen variability. So we send people out flying all over US, traveling, driving around, collecting rust samples from across the US. We call our collaborators, every research station, every university, hey, if you go out there and you see some rust, send us some samples. So we get hundreds of samples on a yearly basis and analyze them. And with oat crown rust, when you look at it, majority of the samples we got from winter wheat, being the dark bar, and the spring wheat are essentially unique. That means they're like humans. There are not that many twins among them. The twins are far in between, and then triplets and quadruplets and all of those, you don't see that much. So what it means is that it's highly variable. Is it due to buckthorn, or is it to the wind that is blowing north? We know that this variability essentially causes the effectiveness of those resistance genes that I mentioned to you to disappear quickly. In US, those resistance genes are about 3 to 5 years maximum, about 3 years. In Australia, it's a little bit less, or about 3 years. In Brazil, it's 0. As soon as they release an oat variety, it's susceptible. So it doesn't matter what resistance genes they put in there, it's going to be susceptible. So the question becomes, how are these pathogens generating so much variability to create that much of a virulence into the crop? So what we have used, and a little bit northwest of here, if you see there's a sign that says Batmore buckthorn plots. And essentially you have these buckthorn hedges growing. And a scientist in our unit, Roger, who always trims them, hates them very much. But essentially what we use them for is to generate that genetic variability. So these buckthorns are being used to generate lots of rust variability. And in the spring, we plant all kind of oat varieties in between. And then what we are looking for is there oat varieties that are resistant to the pathogen or not. So we do this on replicated trials. We do it on multiple years to essentially find oat varieties that can resist the variability of the pathogen that is being generated. So buckthorns are good sometimes. So you can see this is a variety that was recently released by a retired USDA scientist, and hope there is no USDA administrator. We love our job. We even work without paycheck. Essentially, you can see this is a resistant oat variety right here. Next to it is a susceptible sib. resistant, susceptible. This is a gene that was essentially introgressed from a wild oat species into this line. And you can see at maturity, the resistance have some grain on them. But you can see the susceptible ones are just sticks. So you can see how much of an effect crown rust can have on oat yield. So this was recently released. Hopefully it will last more than 3 years, but we don't know. Oops. So what that sexual cycle does on buckthorn is essentially creates multitude of races. All of these races are highly variable. There is no predominant races. In some of the asexual rust, for example, leaf rust and stem rust and stripe rust, you can see a race that is essentially virulent to a number of genes. You don't see that predominance here. A mixture of population that intermates without any problem, and there is no correlation between the virulence phenotype and the genotype. So we essentially have to phenotype every single rust isolate that we get to find out what are the virulence that it has. As I mentioned, there's over 100 plus resistance genes that we know, but bottom line of it is resistance is futile because the lifespan is only 3 to 5 years. So it's not going to last you very long. There is no apparent fitness cost to the pathogen for virulence. And there is no negative association between virulences. So pathogen can have multiple virulence genes infecting multiple resistance genes, thanks to buckthorn. So the question we are asking is, there's a scientist in my unit that is working on this, is sexual reproduction on buckthorn the major source of variability? Or are the asexual wind-borne pathogens have as much genetic variability as the buckthorn? So we have samples that are collected from northern part of the US, the southern part of the US, where there is not much buckthorn known out there, to essentially look at that. What is the contribution of buckthorn to the spread of this disease? I keep mentioning the wind-borne going up north, but are the buckthorns up here contributing to this disease? So if there was no winter wheat grown in southern part of the US, would that stop the pathogen from moving north? Probably not. And what is the genetic variability in buckthorn? That is the part that we are not studying. Are there buckthorn genotypes out there that are essentially resistant to crown rust? Are there buckthorn genotypes out there that are more susceptible to crown rust? In some places, when you walk around, you can see that there are buckthorns that are more red than the one next to them because of all the rust that is being grown on them. What is that value? So are there variation there? We don't know. So in terms of oat, because there are some of you guys that are worried about oat crops that is being grown, what we are essentially working is looking for slow rusting, general type of resistance, not single gene resistance. So we know because single gene resistant that we have used don't last very long. So can we find a happy intermediate? Can we find an oat crop that gets rusty but doesn't get too rusty? So it can produce a grain that we can use. So we are looking at this type of variation to see if we can find that type of oat germplasm to essentially utilize that to breed for a crop that is more tolerant to the crop. But what I want to leave you with and the take-home message is this giant black box that we don't know. Of what is buckthorn doing, what is the variation in buckthorn, and can we essentially find out if studying the buckthorn can help us essentially reduce all of these diseases. So I leave you with this. That's what our hope, that at the end we make this a historic slide so you don't see any rust blowing at the end of the combines. Any questions?
We've got time for a few questions, and I'll try to pass this around so people can hear the question when it's asked.
I hope I got you guys onto coffee break time.
Very interesting. Are you doing all of this through traditional plant breeding?
Yes, sir.
And is there any work being done with any of the gene editing techniques like CRISPR or anything, and what's the future?
There is one group that is working on that in oat, but as you can imagine, there is not lots of funding for that type of research in oat. And people are not interested in genetically modified oat crops. So,
What about genetically modified buckthorn?
Rob, he would know the answer. I don't know if anybody is working on the buckthorn side. As I said, that is, I think, the major black box. We really need to understand the alternate host that can be essentially create genetic variability in buckthorn. Because if you find buckthorn that are very susceptible to rust, that might be a way to eradicate buckthorn. Just plant rust-susceptible buckthorn. Or aphid-susceptible buckthorn.
Or make them susceptible.
Right.
Any other questions here? No. Okay, if not, let's thank Shahryar again, and we'll get ready for our next speaker.